Welcome to the Induced Innate Immune Responses module. And in this talk, we're going to look at the NOD-like receptors. We'll first learn about the structure of NOD-like receptors, followed by the cellular localization and signaling pathways that are initiated by the NOD-like receptors. We will then look at the outcome of these signaling pathways in this talk. Now, some proteins and cells that play critical roles in innate immune defenses are always present in the host body. These include proteins like lysozyme, defensins, or complement proteins. Thus, they are constitutively present. We also have cells like monocytes and neutrophils that play crucial roles in innate immune responses, and they are also always present in the host body. However, when there is infection that occurs, then some proteins have to be made in response to that infection. And thus the expression of these proteins have to be induced. Usually these are proteins that are involved in inflammation. And their expression has to be induced because if these proteins are present all the time, or if there is constitutive expression of these proteins, then it is actually detrimental to the host. Inflammation normally occurs when the host is able to recognize pathogens that are present inside. The role of inflammation is to direct molecules as well as cells to the site of infection, and these molecules and cells will then play an important role in clearing the pathogen. Another role of inflammation is to activate the immune cells so that they do a better job at clearing the pathogen. Inflammation also induces localized blood clotting, and this helps in curbing the spread of the pathogen to other parts of the body. And finally, inflammation also helps in repairing injured host tissues. Now, inflammation is triggered by the production of different molecules like cytokines, chemokines, and other types of mediators like histamine. The host is able to recognize microorganisms that cause infections by recognizing molecular patterns that are present on these pathogens. These patterns are called as pathogen-associated molecular patterns, or PAMPs. The PAMPs are recognized by molecules made by host cells, which are pattern recognition receptors, or PRRs. Now, the pattern recognition receptors are not only able to recognize PAMPs, but they're also able to recognize damage-associated molecular patterns, or DAMPs. And these patterns occur when there is damage to tissues. The pattern recognition receptors can be soluble molecules like phycolin or mannose-binding lectin, but they can also be membrane-bound or intracellular molecules. The pattern recognition receptors that are present in host cells come under different families. Some come under the toll-like receptor or TLR family of proteins, while others come under the NOD-like receptor or NLR family of proteins. We do have pattern recognition receptors that belong to the RIG1 family of proteins, as well as the STING family of proteins. The NOD-like receptors, or NLR family of proteins, are a family of pattern recognition receptors that get their name because of the presence of the nucleotide binding oligomerization domain that is present in all members. Now, there are other domains that are also present in the NOD-like receptors, and based on the type of domains present, the proteins are categorized into different groups. The NLRA family of proteins have members that have the AD domain in addition to the NOD domain, as well as leucine-rich repeats that are shown in those green oval structures. The NLRB family of proteins have BIR domains, in addition to the NOD domains and the leucine-rich repeats. The NLRC family of proteins have members that have a CAR domain in addition to the leucine-rich repeats, whereas some members can have an X domain instead of a CAR domain. The NLRP family of proteins have the PYD domain in addition to the leucine-rich repeats. And thus we can see different types of domains present in the NOD-like receptors. The NOD-like receptors, or NLR proteins, are an ancient family of innate immune receptors. They're not only found in the kingdom Animalia, but homologs of NLR proteins have also been observed in plants. Now, some NLR protein members can activate signaling pathways that induce the production of cytokines, chemokines, and antimicrobial peptides, and this is usually accomplished by activating the transcription factor NF-kappa B. However, there are other NLR members that also produce inflammatory cytokines. However, it is done through another pathway that results in the formation of a complex called as the inflammasome. NOD1 and NOD2 were one of the first NLR proteins that were discovered, and they both have the caspase recruitment domain, or the CAR domain. NOD1 and NOD2 are pattern recognition receptors, and the PAMPs that they recognize are peptidoglycan fragments. Now, peptidoglycan is found as part of the cell wall of bacteria, and they come in normally two flavors. You have the peptidoglycan, where in the stem peptide region, at the third position, a lysine is present. While in other forms of peptidoglycan, at that third position, you have the mesodiaminopimelic acid, or MDAP, present. Peptidoglycan, where lysine is in the third position, is normally found in gram-positive bacteria, while peptidoglycan, where MDAP is present in the third position of the stem peptide, is usually found in gram-negative bacteria. Now, when we look at the lysine-containing peptidoglycan as well as the DAP, or diaminopimelic acid-containing peptidoglycan, in both cases, to the N-acetylmuramic acid, we have the stem peptide attached. And the first two amino acids that are attached to that stem peptide are the alanine and the glutamic acid. Now, this moiety of the N-acetylmuramic acid that is bound to alanine and glutamic acid is also called as muramyl dipeptide, or MDP. This MDP is the ligand for the NOD2 receptor, and thus NOD2 is able to bind to the fragments of peptidoglycan that have the MDP component. Thus, it is able to recognize peptidoglycan fragments from both gram-positive as well as gram-negative organisms. When we look at the NOD1 receptors, we now have to look at portions of the stem peptide, and when we focus on the one that's in the smaller black box, we see a peptide which has two amino acids, which is the glutamic acid and the mesodiaminopimelic acid, or MDAP. This is called as IE-DAP, and it is a ligand for the NOD1 receptor. NOD1 receptor can also recognize a tripeptide, where you have the MDAP that is bound to the glutamic acid as well as to alanine, which is called as TRIDAP. It can also recognize a moiety where you have a tripeptide, which was the TRIDAP, that is linked to N-acetylmuramic acid, and this is called M-TRIDAP. All these different moieties are able to bind to NOD1. Thus, we can see that NOD1 is able to recognize peptidoglycan fragments that have the M, or the mesodiaminopimelic acid. When NOD1 or NOD2 bind to their appropriate peptidoglycan fragments, the NOD signaling pathway gets activated. Now, binding of the ligand to the appropriate NOD receptor results in the dimerization of the NODs, and this dimerization results in the recruitment of a kinase called RIP2. This RIP2 is able to interact with the NOD receptors through their CARD domains. Now, RIP2 is able to recruit an E3 ubiquitin ligase called XIAP or CIAP1-2. And what these ligases do is they get activated and they build a polyubiquitin chain. This polyubiquitin chain can act as a scaffold and recruit other proteins like TAB1 and TAB2. Thus, when TAB1 and TAB2 are bound to ubiquitin, then they also have a kinase associated with them called TAC1. The polyubiquitin chain scaffold can also recruit another complex of proteins, which are the inhibitor of kappa B kinase or IKK complex. This IKK complex can get phosphorylated by activated TAC1, which is also bound to this polyubiquitin scaffold, and thus the phosphorylation of IKK results in the activation of IKK. The activated IKK then phosphorylates the inhibitor of kappa B or I-kappa B, which results in its ubiquitination and subsequent degradation by the proteasome. This allows NF-kappa B to be free, and it is able to enter the nucleus and transcribe genes that usually code for inflammatory cytokines. Some NOD-like receptors like NLRP3 are able to form a multi-protein complex in response to an infection, and this multi-protein complex is called as an inflammasome. Now, similar to NOD1 and NOD2, NLRP3 is normally present in an inactive state. However, it can be activated due to events like loss of intracellular potassium ions or generation of reactive oxygen species or the disruption of lysosomes, all of which can happen when there is an infection. And thus here we can see in the figure the inactive form of NLRP3. When NLRP3 gets activated, many NLRP3 molecules are able to come together to form aggregates, and they're able to interact with one another through their leucine-rich repeats domain. Additionally, the pyrene domains of NLRP3 are able to interact with another protein called ASC. ASC functions as an adapter protein, and the NLRP3 aggregates are able to interact with many ASC protein molecules, and thus we have the formation of a multi-protein complex. The recruitment of different ASC molecules results in the formation of a polymeric ASC filament, and this polymeric ASC filament is able to recruit the protease procaspase 1 through CARD domain interactions. Now, procaspase 1, when it's recruited, there are many molecules of it recruited, and thus we see aggregation of procaspase 1. This aggregation of procaspase 1 results in autocleavage, which means it's able to cleave itself to form the activated form of caspase 1, which is a proteolytic enzyme. Once caspase 1 is activated, it has the ability to cleave these pro-inflammatory cytokines that are present in the cytoplasm. However, they're present in an inactive state and hence are called as proproteins. Cleavage of these proproteins by caspase 1 results in the formation of the active cytokines, and examples of such cytokines are IL-1 beta and IL-18. These active cytokines can then be secreted out of the cell, and thus we can see the production of pro-inflammatory cytokines. Additionally, activated caspase 1 can also trigger cell death if the need arises by a process that is called as pyroptosis. With this, we come to the end of our talk, where we learned about the domain structure and cellular localization of the NOD-like receptors, as well as the outcome of the signaling pathways that are initiated by NOD-like receptors.